This lecture is going to be about evaluating treatments in biomedical research. So the aims are to understand the importance of evaluating treatments in medicine and to understand the context and implications of evaluating treatments. We also want to understand the need for randomized controlled trials, and finally learning to critically evaluate randomized controlled trials and understand their main features, strengths, and potential weaknesses. So how do we define treatment in health research, in global health research, in medical research? Well, in most cases, people will think of simply new drugs or medicines. But this can be much, much broader than drugs. We can be talking about new surgical procedures, new medical equipment, new approach to counseling after psychiatric or psychological trauma, new approach to motivate care-seeking behavior, and even vaccination effectiveness and screening programs, risk factor withdrawal, and even we can extend this to look and evaluate different organizations or even entire healthcare systems. So all of this can be evaluated in terms of their effectiveness in reduction of the burden of disease in the population. You know, drugs can be effective, but surgical procedures can be effective. Introduction of equipment can be effective in reducing the burden. Counseling can be effective in mitigating the effects of trauma. Motivating care-seeking behavior can reduce the burden of mortality because mothers are suddenly bringing their children to doctors, so they're not dying at home. Vaccination is a preventive measure. It's not really a treatment, but it can be effective because it can still reduce the burden, and so can screening programs and risk factor withdrawal, anti-alcoholic programs, withdrawal of smoking. And finally, the organizations and their existence can have effect on the burden of disease in the population. And finally, really the entire healthcare system can be evaluated and compared to other healthcare systems in other countries. So it's remarkable how much space and room is there for doing research on evaluating all these interventions, as we call them, under one huge umbrella to reduce the burden of disease in the population. How did the medicine advance to this level? It was precisely through evaluating everything that we were doing all the time to reduce the disease. Because imagine early humans, they would have disease and they would not know what to do. But clearly, whoever has a disease is trying to help themselves. So they were trying all sorts of things, grasses, swimming in cold water, or this or that, anything that would come to mind that could perhaps alleviate the symptoms and make them feel slightly better. So you can imagine what the first surgeries must have looked like. Those were performed without anesthesia and without antisepsis. So they were, unfortunately, people had their legs cut or even breasts cut or stuff without any anesthesia when they had cancer or a wound or injury. And never mind that the surgery being performed in that way was a horrific experience and intervention, but many times the surgeries would go quite well actually, but because of the post-operative care and wound infections, people would still die in vast majority of cases following the surgery, even when the surgery went well. So there was a lot of thought of how to improve surgical procedures, and one major progress in medicine was when James Young Simpson, who actually worked here in Edinburgh, when he introduced anesthesia, and this is still regarded as one of the biggest discoveries that assisted medicine. We don't really even fully understand how it works, because it shuts the body down so that it can't feel anything. It shuts the conscience down, and but it does work. It keeps the patient in this state so that we can perform things that we could never perform under normal circumstances. with patient being steady. And clearly, this was a massive progress. But in addition to anesthesia, just as important, if not a lot more for the survival of those patients who were treated, was Lister, who again worked here in Edinburgh and developed and introduced antisepsis. So thanks to those two massive progresses, after the surgery was completed, the patients would no longer be dying from post-operative infections. And you can see how from these days and these days the surgery then advanced. Antisepsis included all sorts of procedures such as surgical gloves and masks and hats. And finally we got to the stage where we had these modern equipped high technological surgery wards where all sorts of things can be done today, even with robotic surgery, so that we don't even need to worry about the surgeons, how much sleep he got or she got, and or human error. We can just standardize how surgery is performed by programming a robot to do it. So evaluating treatments all over the world didn't have much meaning unless there was a massive uptake of an intervention that seemed to be working to reduce the burden of disease. So this is where the journals, the medical journals, scientific journals, had huge importance, because whoever made a progress and realized that something has worked quite well, they were then almost obliged to publish this so that everyone else, the doctors in other countries and places in their own country, can actually follow this procedure and do the same. So these journals developed as their own field of medicine, the medical publishing, and there is a logic to them, there are rules to them, guidelines, and they always need to be peer-reviewed so that you can't just publish whatever without someone who really knows about this can check it for plausibility and whether this is likely to work or not. So this is why we have a peer review system. It takes some experts to review your paper and judge it before it can be published, to serve as gatekeepers against protecting the human knowledge, medical knowledge, against some completely ridiculous ideas. Because the demand on effective intervention is always there, and it's huge, and if someone publishes something that is not likely to work, that can't possibly be a good thing. Now, the journals proliferated so much, just as did the biomedical research. There's just so many fields of research focused on specific diseases or parts of medicine, and the parts of molecular medicine or biomedicine, that it's very difficult to keep up with that. I mean, there are thousands and thousands of relevant journals today, and it is not possible to follow what they're all saying all the time. So someone was saying for a long time that you should publish or perish. If you don't publish what you're doing, no one's going to know about it and no one's going to uptake it, so you will basically perish. But soon this became publish in a visible journal or perish, because no one can read all the journals that are out there anymore. So what are the visible journals in medical field? Well, New England Journal of Medicine is an American journal which has huge standing. The Lancet is here in the UK, Journal of American Medical Association. Those three journals would be the first obvious thought when you're talking about highly visible medical journals. If you're managing to publish anything in the New England Journal of Medicine or the Lancet or JAMA, that means that you must be among the best in the world and most respected experts in the world in those fields. So there is something about these treatments that is making them very specific in the place of biomedicine generally, which is that unlike a lot of research that is being conducted simply to generate new knowledge, the research to develop new treatments can also be phenomenally lucrative activity, because it can be linked to intellectual property and it can become a very big business. So the big pharmaceutical industries and companies have developed to constantly do research and development to bring up new treatments, and then to offer those treatments to the public and sell them basically. So they can make enormous amounts of profits, pharmaceutical industry, from this activity. And clearly they have to be regulated, because the incentives for people to buy are just their thinking that they're going to be helped through buying these treatments. But in fact, it is sometimes questionable whether these drugs work or not. So there have to be many gatekeepers, because there is a huge opportunity here to make a large sum of money for a company for just exploiting other people's illnesses, and nobody really wants that. So it's easier said than done, because the overall context is as such. You have pharmaceutical companies. Now, one good thing is that they're competing with each other. So if they were not competing with each other, then any company could set price tag for any medicine that they want, and then you could have ridiculously high prices, and people would still be paying for it because they are sick and they need this, or the governments would need to pay for it. So luckily there are many pharmaceutical companies, so through competition they cannot just push prices up, because somebody will undercut their price, and this is going to keep the prices at some more reasonable level. And in all fairness to pharmaceutical industry, we have to be fair here. They are investing enormous amount of their profits, which they acquire on the open market, into research and development. So it's not just academia that is doing biomedical research. The research into development of new drugs and treatments is phenomenally developed within the pharmaceutical industry. And look at the amounts of their own profits that they are happy to reinvest into research and development. Roche, $9 billion a year from their profits goes into further research and development spending. Pfizer, more than 7, Novartis, about 7, Johnson & Johnson. So look at these billions and billions and billions of dollars which companies take out from their profits, and although they could pay this to themselves as bonuses or to their shareholders as dividends, they don't do this. They take it and they invest in further research and development. Why do they do this? Well, again, it's because of the competition. They cannot afford to fall behind, because it's so difficult to get a drug through the process of approval and development and out in the market, that once it's out in the market, it becomes a blockbuster. It makes enormous profit for the company for a limited period of time. But companies are so dependent on their blockbusters that if there is another company which is happy to invest massively in research and development, and that could develop another blockbuster that could kick the present one out, this could just mean the end of the company if they lose their key products due to competitors. So no company can afford to not invest in research and development, because soon enough they may be their key products, best-selling products, best-selling drugs could be kicked out by their competitors who did invest in this. So you can see how a liberal market and the competition between these companies works to the benefit of the customers, because the prices, because of the competition, need to be kept to some reasonable level, and a lot of profits need to be reinvested so that in research and development, so that no company loses ground on any other company. And this is all serving us all, right? So we have to be aware of this. And then you have pharmaceutical industry then putting many, many drugs on the market. What is happening on the other end? So this is the supply side. What is happening on the demand side? On the demand side, you have people who are aging, who are getting more and more sick, and every time they get sick, they believe that they should be immediately assisted and helped. And by whom? By their own government. And why? Because from their income, they were paying tax. And they were paying tax to the government all these years, so they didn't take their money that they earned, they paid part of it as a tax to the government. And now this tax is used by the government to, and they expect the government for this money to then treat them when they're sick. So they expect the government to pay for these drugs. And the demand is huge. They would like all the drugs all the time. Every time they're sick, they would like to be helped. So then there is a government, right? And the government needs to serve as some sort of a bridge between the desires of pharmaceutical industry to sell as many drugs as possible and make as much profit as possible, given how much effort does it take to develop these drugs and how much they invest in them. So they want as many of their drugs to be paid by the governments to deliver to people, and people want as many drugs to be paid by the government as deliver to them. But the government doesn't have an endless budget, so the government needs to be a buffer in between the demand and the supply side and decide how much they can really afford and what drugs are they going to be subsidizing or paying in full, and what drugs will people have to be paying themselves if they're not essential. And this is a constant struggle. And then there are further gatekeepers. So Food and Drug Administration is a very, very strict committee, which clearly there is a huge pressure on them to approve drugs. They are the ones approving drugs for the global markets as safe and effective. If they don't think the drug is effective or safe, that it has if they think it has side effects, they're not going to approve it. And then, so many drugs come through stages, and then stage 1, 2, 3 clinical trials, and then they just don't get approved by the FDA because they don't think the drug is effective or that the drug is safe. It's very difficult to get their approval, because if they do approve drugs, it immediately means there is another blockbuster and it's going to make huge profits for the pharmaceutical industry. Then there is the World Health Organization, who are publishing their guidelines and what they endorse and what they don't. There are researchers themselves who are conducting clinical trials on multiple drugs, and they have to be fair and square in what they're doing. They shouldn't be influenced by pharmaceutical industry to make the results nicer than they are. Many of you will be in a position to test drugs and conduct clinical trials. And finally, there are medical journals who should not also come under influence of the companies to publish research on the drugs which is showing that the drugs are doing really well, without rigorously scrutinizing the designs of those studies and whether they're really showing what they're telling they're showing. So these are the gatekeepers between the huge demand and supply that is hugely eager to sell as many drugs as possible. And there is obviously all sorts of incentives and possibilities in all this to fall under undue influences of the pharmaceutical industry, who are interested in getting their drugs approved. So we need to ensure that the governments are up to their job, that Food and Drug Administration is rigorous and strict, World Health Organization as well, researchers involved in testing drugs too, and finally the journals need to monitor closely what they're publishing as well and make sure that it's a rigorous research. So obviously, evaluating treatments requires the highest standard of research. It requires evidence beyond reasonable doubt, requires some sense of responsibility and very high moral standards from researchers who evaluate treatment, editors of the journals that publish this research, members of Food and Drug Administration and World Health Organization or other important advising bodies, governments and their committees on health care, and corruption is clearly a real concern at each stage, given all the stakes and multi-billion-dollar profits that are possible. Clearly, we should start with thinking about what evidence can we gather that some treatments are effective. Anecdotal evidence should always be dismissed as completely useless. If a grandmother tells her granddaughter, "Don't sing while eating your soup because you're going to marry a crazy husband," that's superstition. We clearly won't think about it as evidence. If mother tells her daughter, "You seem to be coming down with something, take 3 vitamin C pills," that's probably something she overheard from her friend or read in some daily newspaper, but this is not evidence. What evidence do we have that these work? So again, if you overheard on a ward that a doctor gave a drug to a patient last week and she did remarkably well, so let's just give that drug to the new patient. This is one single case. You cannot possibly think that if something worked in one single case, that it's going to work for everyone. So we shouldn't base recommendations on medical treatments of this kind of evidence. So let me try to explain an example of evaluating a new treatment. Let's say there is a new chemotherapeutic agent for ovarian cancer, which was just recently approved for testing by Food and Drug Administration. I'm going to try to explain to you what does it take to actually develop a blockbuster drug by a pharmaceutical industry, so that you can understand a bit better just how difficult this is. So let's say that it took 7 years of research first, which needed to be funded, to find a new target mechanism in some types of ovarian cancer that was not known before. After this, it took 3 more years to develop and chemically produce biologically active substance in sufficiently large quantities. After this, you need to take 4 more years to establish both the effectiveness and safety in mice with induced ovarian cancer, for example. Then you need further 5 years to study safety and toxicity in humans and to calibrate the correct dosage. And finally, look at all these 7 years and 3, that's 10, 14, 19 years. So it takes 19 years and perhaps $300 million to this stage to perhaps get a projected revenue of $150 million a year in the United States and European Union. So, but we're not done here yet at all. So we are only here at the stage where we can actually try to test this on humans, whether it works. So far we have only been developing something that could have potentially worked, producing it chemically in sufficiently large quantities, establishing whether it's effective in experimental animals, and then checking whether it's safe for humans to even administer. Now that we've established that, now we start looking at whether it's actually effective in treating diseases, all right? So let's say that you're a research team leader at the New Royal Infirmary here in Edinburgh, and you expect to see 70 new cases of ovarian cancer in the following year. To remove the effect of the progression of disease, you only choose 30 cases that are admitted with stage 2. So now we are actually finally looking at, is this going to be effective in reducing the mortality from ovarian cancer? Remember, there already are plenty of drugs for ovarian cancer. So not only that we need to establish that this is effective against ovarian cancer while it's not toxic, but also we need to show that it's more effective than the existing treatments that are already there. And this is why it's so incredibly difficult to launch new drugs on the market. Because it's a bit like, you know, if you had a boyfriend or a girlfriend, you have one boyfriend and then you see he's horrible, and then you have another and it's better than the first one but not much, all right? And then you have the third one and the fourth one, and they're just getting from bad to worse, really. And after trying 7 or 8, you realize that that's really as good as boyfriends get, and you really need, you know, some miracle to push beyond that standard, and then you go back to perhaps number 2. It's the same with the drugs. You know, nature is extremely complicated and we don't know how anything in organism works, so we're trying to find the drugs that work, and none of them actually works perfectly and deals with all the problem in everyone. That's very, very difficult to find such a drug. So basically what we're trying is combining the drugs that exist and their strengths, and at some point you reduce 50%, 60%, 70% of the problem. Once you reduce 70% of the problem, it kind of becomes the law of diminishing returns. It becomes extremely difficult to think of something that is going to outperform everything that we already collectively found to that point. So now we are testing this drug to see whether this drug perhaps could, all right? So we firstly need to remove the effect of the progression of disease, because we can't compare 2 different drugs if we are performing or administering one drug to advanced stage of the disease and the other drug to non-advanced or early stage. Clearly, those in early stage will have better prognosis no matter what, whether they're treated or not, in comparison to these in late stage. So this is why firstly we are trying to remove the effect of the progression of disease, and we're going to only choose 30 cases that are admitted with stage 2. Okay, so not stage 1, not early, but not stage 3 either, not too late, or 4. Now we're going to randomly assign those 30 patients into 3 groups, and each group will receive different treatment. Only one will receive chemotherapy in addition to an already acceptable treatment. Now we're going to follow them up for 5 years, and we're going to compare their survival rate, okay? But note, this is another 1 year and 1 year to collect all these cases, and then 5 years of follow-up. So this is another 6 years of time, and you remember we already used 19 years to get here, and now we need 6 more years to collect the patients and then to test the treatment. Now, why do we need to randomize patients into 3 groups? They need to be randomized, you will remember, because randomized controlled trials get rid of all the background effects, all the confounding effects, all the other effects that are different in these 3 groups, apart from the treatment itself. So our intention is to minimize the effect of anything else by purely randomizing the group into 3 subgroups, and then we hope that all the other factors that could contribute to survival will also be randomly assigned, meaning equal between 3 groups, and the only effect will be attributable to the treatment itself in the cohort of patients which are of similar progression of disease. Then we are kind of comparing like with like. The only question is number 30 of them enough? Let's see what happens. Let's say that this is what happened. 1, 2, 3, 4, 5 years of follow-up. This is what happened. So, survival in stage 2 ovarian cancer, 30 patients. This group had a radical surgery with this new chemotherapy, but you only had 10 of them, right? Look, they survived, 50% of them survived, 50% of them unfortunately didn't. This group had radical surgery, no chemotherapy, and this group was left untreated. Look at how poor survival rate is there, only less than 10% really, right? Now, anything strange about all this? Well, if radical surgery was the only thing that we could do, and now we have a chemotherapy, this is how we should do it. But were we allowed to leave anyone untreated? Is this acceptable? No, it's not acceptable. Okay, so we can't have this arm which is untreated. As long as we have a treatment which is effective, we can't then have an arm of untreated. Today we only have untreated as historic cohorts for comparisons, but we do not leave anyone untreated in trials anymore. Okay, so you have to provide the control group the best possible treatment at the time, and then also there is another ethical issue. When do you have to stop the trial? Because sometimes you're going to have to stop the trial. You're going to have to stop the trial if very soon or soon enough or before, anytime before the end of the trial, the difference between the treated group with the new agent and that with the best standard up to that point becomes so large that you can establish that it's statistically significant. At that point, you're no longer ethically allowed to continue your trial. You have to stop it, and you have to provide everyone this treatment. So you can see that at this point here already, but perhaps particularly here, there was a wide gap in survival, 55 versus 30%. At that point, probably you would want to stop the trial if the significance was reached. And this is an example why you need statistics, because you need to know whether this is significantly different or this is perhaps just purely a chance effect and could have been the other way around. So how do we know this? Well, let's first go back and think about randomization before we get to the uncertainty of it. There are some methods through which you can randomize. You can do it haphazardly in an undescribed way, which is totally unacceptable. You can toss the coin or draw the cards. That's okay, but not confirmable really later or replicable. So it's good to use pre-randomized lists, but the trouble is that the entry bias is possible here for some reason that is difficult to account for. Now you could use sealed envelopes and just assign randomly numbers to people after trying to randomize those envelopes, and you can use this, but the trouble is that then the patients themselves could swap the envelopes for some reason which is again beyond your understanding. So You can identically package treatments, that's recommended if feasible. You can randomize by telephone, which is recommended, and then you can randomize by computer, which is very highly recommended. In this day and age, you really want to randomize those 30 people using computer randomization process, and then you just basically you are fine. I mean, these telephone randomizations were used before. You would have random number tables, and then you would be looking at a certain number that comes next, and whoever has that number in their telephone number, you would pick that person. So that was kind of a random process of who gets assigned where. But nowadays, why bother when you can actually randomize by computer? Let's go back to our example, all right? So we would like to be sure that the observed differences in survival were indeed due to treatment and not to any other factors that could have influenced survival. This is why we randomize. Now, there are two types of error which need handling in randomized controlled trials still. The first one is random variability. The second one is systematic bias. And you need to understand which error are you handling through which procedure, okay? Now, let us explore the characteristics of the three randomized groups. You remember the group A was left untreated. Only one person survived. Mean age of this group was 65.6 years. This was the grade of malignancy, another very important risk factor for survival. Mean number of other concomitant diseases, another important risk factor, and then smoking. Again, you see 50% of them smoked. Now look at the survival of group B, which had surgery, and group C, which had surgery plus chemotherapy. So clearly there seems to be a difference in survival, positive for this group that received chemo. But look at this. At the same time, this group of untreated was also the oldest group. This group was much younger when they were admitted to treatment. Histologic rate of malignancy, again, this group has the most favorable group C, much more than, for example, group B, which looked very bad. And mean number of other diseases, because you only had 10 people in each group, there is, again, quite a big difference, actually. 1.5 is a lot less than 2.6. And finally, look at the difference in smoking. There is some difference, too. So, although we tried to randomize in order to make all of these background risk factors similar, they're still different, and we still can't be sure how much these other things influence survival rather than the chemotherapy itself. So the groups were not comparable after randomization. And already because of this, you need to say and conclude that this trial is inconclusive, because you cannot rule out that it was really these other factors that influence survival rather than the treatment itself. And then there is a problem of chance effects. When you have small sample sizes, then you have phenomenal chance of random variability. To illustrate this case very simply, if you roll the dice, you can get 1, 2, 3, 4, 5, and 6. If you roll the dice 10 times, you can record the distribution of 1s, 2s, 3s, 4s, 5s, and 6s. And you can have something like this. 3 times you get 6, 1 times you get 5, 2 times you get 4. That's completely possible. But then if you do it again, it's also completely possible to get something like this. 0 times 2, 2 times 1, 2 times 3. And then third time you get 4 times 4, and 3 times 5, and 0 times 3. So you can see that these are very, very different. Although they are random, each one of them is random, but they are very different results. But what if you roll the dice 3 times 1,000 times? This is what you'll probably get, or something extremely close to this. You're going to get practically the same number of each number coming up after 1,000 rolls of the dice. So this is why we're doing randomization, because if we have large enough sample sizes and we properly randomize, then we really believe that everything in the background that could be a confounding effect or a bias will actually be leveled between three groups, and the only difference that will remain will be the treatment they receive. So this is the visualization of the reason why we randomize, but it's also a visualization of why the power of the study is so important, and why the size of the sample is an answer to the problem of chance. You just, in epidemiology, bigger really is always better. The bigger the sample, the lesser the chance effects and their influence of your results. So you should learn from this that we can virtually eliminate random variability with sufficient sample size. If you have large enough sample sizes, you are less and less and less worried about random effects in your sample. Biases, we have to still control for. Confounding, we are trying to control for with randomization. And remember, random effects, we are controlling not through randomization, but through increased sample size. Chance is controlled by sample size. So in epidemiology, we're always worried in concluding various things, whether chance could have an effect, whether biases could have an effect, and whether confounding effects could have had an effect. So remember, through randomization, we get rid of confounding effects as much as possible, also some biases as well, but through the solution to chance effects is really the increase in the sample size. Okay? Good. Now, what if randomization worked spectacularly well and all three groups had similar characteristics? Could we then conclude with certainty that the new treatment is definitely better? So we're going back to the first example of only 30 people looked at. And what if we're so lucky that, look, this is what happened after randomization. Mean age was exactly the same almost. Histologic rate of malignancy the same. Mean number of other diseases the same. Smoking the same. So this is what happens when randomization really works. And survival is still different. What would happen? Then you can test whether these differences are actually statistically significant. How uncertain or certain can you be that the difference of between 50% and 20% is actually statistically significant difference, or just still possibly a chance effect? So this is where statistics come handy, because it gives you some sort of confidence intervals, and this is just an example of what they could be on such a small sample size. Even if you get 10%, this still may mean that if you had a much larger sample, they could be as low as this or as high as 56%. So drawing, this is like tossing the coin 10 times. You may get completely the deviation from 50%, or rolling the dice 10 times, you may get a very big deviation from what you will eventually see. So we are still not certain, and you can see here as well that these confidence intervals all overlap and contain each other, and as long as they do, if they're like 95% confidence intervals or 99% confidence intervals, as long as they overlap, you cannot conclude with sufficient likelihood that those are different results, all right? So you may wish to use 90% confidence intervals if you want to be 90% sure, 99% confidence intervals, 99.9% confidence intervals, and so on. So p-value here is going to be, there's still 27% chance that they are not different, even though the randomization works spectacularly well. So this is telling you that with small sample sizes, you actually cannot achieve statistical significance, even if you see a very big difference. Even if you saw 10% and 10% and 90% here, it is questionable whether you could still conclude based on 10 cases in each group that these are statistically significant. Maybe you wouldn't be able to reach the power of the study. This is what we call statistical power. The study is underpowered. Even if you see the real effect, you still cannot conclude with certainty, or at least high level of likelihood, that these are sufficiently different. So the study is underpowered and confidence intervals are broad, and the solution to this is, let's take a larger study. Let's really increase the sample size. So let's say that now, instead of 10 people in each group, we had 1,000 people in each group. So altogether 3,000, and we got exactly the same results: 50%, 20%, 10%, okay? Now, let's see, how did we get so many people? Because we conducted a large multi-center trial in 17 different countries, which involved 3,000 patients in stage 2. And this is why many of these big pharma trials all over the world involve multi-center studies in many hospitals all over the world, because it just takes time to acquire them. So this is a way to speed up the process and also to get very large sample sizes and be certain. And look what happens here now. If we properly randomize them, then the mean age becomes very similar, the histologic rate as well, mean number of diseases too, and look at how much more narrow the confidence intervals now are. If you see 10% in 1,000 people, you then know that this must be somewhere between 7 and 12%, but it can't possibly be 20 or 50%, and similar for 20 and 50, and p-value becomes very low. These are just indicative, they may not be actually correct if you use the procedures, but this is just to explain to you what really happens if you actually, how these confidence intervals begin to narrow, all right? So this means that there is a less than 1 in 10,000 chance now that there is absolutely no difference between group B and group C. So the study is clearly adequately powered and confidence intervals are narrow. As a result, we have a trustworthy trial. But even so, what could still potentially invalidate the results of that trial? No matter how good it looks, always you have to remain skeptical as a scientist, because the opportunities and incentives to cheat in these kind of trials are large, as we explained. We don't want to accuse anyone of anything. Obviously, everyone is innocent until you prove that they've done something wrong, and that is the right way to look at it. But there are such incentives that you should really remain careful. So, for example, the things that did happen in the past and were discovered was that all the survivals within the group receiving chemotherapy could have been concentrated in the same hospital or country. That is a little bit suspicious, because if you had 20 different countries and you look into those who really survived spectacularly, and they were not in France or Scotland or New Zealand, but they may have been in some other countries, all of them focused, or even in one country, and this skewed the results. That is strange. I'm not saying anything wrong has happened, but that is strange. Now, did the analysts or statisticians know which patient was allocated to which group as they were receiving? Because at the end of the day, all these results end up in the hands of one person, and that one person needs to be completely blind to who is a group A, group B, and group C. That person cannot possibly know or shouldn't know who was in which group, and they should just deliver the results as they are to those who are conducting the trial. This is why statisticians should always be someone completely excluded and kept out of this study, all right, blind to this information. Who was actually sending this information to the statistician? They needed to be blinded to treatment received. And was it possible to ensure blinding of researchers and patients? Because ideally, you don't even want the researchers to know whether they are giving out the treatment or just placebo. Patients shouldn't know either, so that you eliminate any placebo effects that could be positive from them. So, what do we need? We need large sample sizes. We need randomization, we need blinding. Those are the ways to prevent most of the problems in randomized controlled trials. Large sample size will get rid of the chance effects. Randomization is going to try to control for confounding effects and various biases. Blinding is really to control for biases. So in these 3 ways, you control for the things that we're really worried about, okay? So large sample size will also ensure sufficient statistical power to detect and properly recognize true differences, even when they're relatively small between groups. Because the problem in epidemiology many times is that you detect a real effect, but it's very small. And then it's very difficult to actually prove that effect. This is the limits. This is where epidemiology reaches its limits. You can design the study beautifully. You can have large sample sizes. You can control for confounding and bias, randomize. But what if the effect is only 3 or 4%. You need enormous sample sizes, and you need to be phenomenally sure of each step of the way that you've taken, that you've not didn't make any mistakes or didn't do anything wrong, if you are really to prove such a tiny difference with certainty, okay, with certainty of 99.99%, to prove the tiny difference of 3% in treatment results with all the chaos and all the other effects that could have caused it is extremely difficult. So this is where epidemiology really approaches its limits. There may be many tiny effects that epidemiology just cannot prove, because we can't develop a study that would be that big and well conducted to make sure that this is achievable without the confidence intervals around the two measurements not touching themselves. If they're touching themselves, then it's you can't conclude that they're different with certainty, all right? Okay, so randomization will ensure that no systematic differences between study groups exist, and that the disease outcome is mainly attributable to the treatment, and blinding protects us from the results of randomized controlled trials, from all conscious biases and even unconscious biases, which also do exist and can be very, very important. Because of all these problems that we need to think about in conducting randomized controlled trials and evaluating treatments, it's good to do it using some guideline or algorithm or checklist, because it's very difficult to remember every little thing, and very easy to forget to do something, and that can cause problems. These guidelines were really firstly developed in air traffic industry, because the pilots needed to have guidelines, because you could not leave it to a pilot or a crew to think that they remember to do everything before they start flying. It was too risky, because they could have had a bad night's sleep or being distracted with something on the day and so on. So nothing is given to chance. If you're a pilot, you have to go through your checklist so that we can ensure that everyone always does everything they need to do. In a similar way, the people who conduct randomized controlled trials should really behave similarly to those pilots, because this is such an important exercise that has implications for lives of many patients, for their safety, for their survival. So we can't just leave it to chance, and it's good to know and remember, has this been ethical? Was there a sufficient statistical power to detect clinically important difference? You need to account for the patient dropout, because over time you may account that too, so end up with a smaller number of patients. Was the allocation truly random? Did you check for comparability of groups? Were there any co-interventions in different groups? Any compliance issues of patients? And then, was everyone in the study truly and fully blinded? Because they are not supposed to know. The minute they know, you can start having all sorts of conscious, unconscious biases and prejudices, and things may go wrong. Okay, so this is how we should evaluate treatments in medicine. This is a big field of research, very exciting. Many of you will be actually conducting clinical trials, so remember that you need absolute rigor, high moral standards, and because this is a very, very responsible activity for a medical doctor and a researcher. Okay, so I think we are finished with this topic, and we're going to now move on to the next. Thank you very much.